Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar on enabling new cell therapies with GMP-compliant multiparametric cell sorting. I'm Elisa Manzotti, founder of BioInsights, and I'm delighted to be joined today by leading experts from across the sector who will be sharing their experiences and advice regarding this crucial manufacturing step. But first up, we will have a presentation by Dr. Felix Epler, Global Product Manager for Cell Sorting at Miltenyi Biotec. Before joining Miltenyi Biotec as Global Product Manager for the MACSQuant Tyto Cell Sorting System in 2018, Felix worked as a research associate at the Limes Institute, University of Bonn in Germany, with a very strong focus on molecular immunology and cell biology. And following his presentation, we'll be joined by our three panelists for today's Q&A session. As always, do feel free to post your questions to our panel using the Ask a Question box, and we'll try to get to it during this session. But for now, I will hand over to Felix for today's presentation.
Thank you very much for that kind introduction, Elisa. Also welcome from my side to today's roundtable discussion here on Cell and Gene Therapy Insights. The topic we want to discuss is how to be enabled to new cell therapies with GMP-compliant multiparametric cell sorting. My name is Felix Epler. I'm Global Product Manager for MACSQuant Tyto Cell Sorter here at Miltenyi Biotec. And I would like to give you a short introduction into that system and also to give you an application example from the field. So before I start, I think it's important to ask the question actually, what is the motivation on bringing multiparametric cell sorting into clinics, into clinical applications? And of course, currently we are in an era of development of new and innovative cellular therapies. And those therapies are, of course, mainly based on specific cell types or even cellular subsets down to antigen specificities. So it is important to isolate and enrich those specific cells, mostly from heterogeneous cell populations. And this process often is based on using multiple parameters in order to do so. Now, with that, of course, there come also some challenges, and that is there's a lot of instruments currently available in the field which easily can do that job, but actually they were predominantly designed for research applications and not considered to be applied in clinical applications. And this is also why a translation of respective research protocols into the clinics often is very difficult due to, for example, open systems which are prone for contaminations, but also the absence of exchangeable tubings and fluidics, which come with the problem of sample-to-sample carryover, or also the sorting technology itself, which might be very harsh and in the end does not lead to functional or viable cells in the end. And here at Miltenyi Biotec, we tried to tackle those problems by developing the MACSQuant Tyto Cell Sorter. And in the beginning of my talk, I would like to give you a short technical introduction into the system. And in the second half of the talk, I would like to give you an application example on an ongoing clinical trial in the field. So coming to the first part of my presentation, the technical introduction. So the MACSQuant Tyto Cell Sorter, as you can see here in those pictures, is a benchtop cell sorter instrument. Cell sorters are often quite big. This one is a benchtop instrument. You can see it on the upper picture being placed in the CHUM Super Hospital. The Canadian press directly gave it the nickname cancer fighting machine. It's actually quite an interesting video from breakfast television. You can watch that on YouTube. And what you can see is that we have two screens on the system, one big that you can easily see all your gates and subpopulations and work with a software. We have also an internal touchscreen monitor. The whole system is covered with a metal sheet housing, and we have a chamber where you can implement your sample of interest, and this is also giving a control temperature, so you can define a sorting temperature between 4 and 25 degrees of Celsius. Concerning the optics, we are talking about multiparametric cell sorting. Of course, this is also true for the MACSQuant Tyto. We have 3 lasers in the system and can sort based on up to 10 parameters, 8 fluorescent channels as well as 2 scatter channels. This already gives quite a big experimental flexibility. And due to time reasons, I don't want to go into too much technical specifications on the instrument itself. But of course, you can find more information on our website, for example, in the user manual, or you can contact us at Miltenyi Biotec if you're interested in more detailed information. Where I would like to go into more detail is actually the sorting principle itself, and what you can see here is the MACSQuant Tyto cartridge. So the Tyto is a cartridge-based cell sorting system, and this of course comes with some benefits. For example, that the cells won't leave this cartridge during the sorting process, so are always in a closed and sterile environment. You can see this cartridge here has 3 different chambers. An input chamber, you can see that on the left picture, the left chamber. There's also a mixing propeller inside, preventing the cells from settling down. And this is actually where you put in your sample of interest. Then in the middle, we have a positive collection chamber where the positively sorted cells will be collected. And on the right side, a negative collection chamber. And here, everything else flows through. There's no waste, so you won't lose any cells, and you can always regain your cells from the cartridge and start another sorting process. On the bottom of the cartridge, there's a microchip. And on the right-hand side, from the bottom view, you can see that much better. This microchip is actually the heart of the whole system. So here, the cell analysis as well as the cell sorting process takes place. And it's really, really small. So you can see that also with this comparison. But if we zoom in a little bit, we can see that there is a channel, for example, so the cells come from the left-hand side on the lower part, run through that channel, pass the lasers, are analyzed, and in case of a positive event, they are sorted into that upper channel. All the rest just simply flows straight ahead into the negative collection chamber. And the sorting process itself, we can have a look at that, is mediated by a microvalve. This microvalve can be opened magnetically. This is a very fast process, only taking around 30 microseconds. So the microvalve can move up to 30,000 times per second. And it simply redirects the positively recognized cells into another channel, a very gentle process. So the cells will end up in the positive collection chamber. All the rest simply flows through straight ahead into the negative collection chamber. And if we now have a look on the whole cartridge during the sorting process, we see that the only thing coming in and getting out of that cartridge is sterile filtered air. It enters at the bottom, coming from the instrument, through an 0.1 micrometer filter, then enters the input chamber through another 0.1 micrometer filter, builds up a very, very mild air pressure, brings the cell suspension over the microchip where analysis and sorting takes place, and the positively cells then end up in the middle compartment, and all the rest simply flows through to the negative collection chamber. And that is actually how the sorting process itself is really working. And you can already see it's really different from what we know from most of the systems out in the field, the so-called FACS droplet sorters. You can see here a comparison to both of the technologies. On the left-hand side, the FACS droplet sorter. In this system, cells are actually set into small droplets, which then leave the system, are electrically charged and redirected in an electric field in order to be collected in different collection vessels. You can already see there are several problems. So those FACS droplet sorters are actually open systems, so there is a high risk of contamination. The fluidics and tubings of most of these systems are fixed inside the instruments and therefore shared between different samples, which has a high risk for sample-to-sample carryover. And those systems work with very high pressures, followed by a strong decompression when those droplets leave the nozzle. They're directly electrically charged, then hitting the collection vessels with quite high shear forces, so it's a stressful sorting process, which might affect also cellular functionality. In contrast to that, with a MACSQuant Tyto, you have a very gentle sorting process. It works with a very low pressure, therefore there's no strong decompression, also no charges applied to the cells, and only mild shear forces act on the cells. But in addition to that, it's also a very safe cell sorter. It's, as I said, working in a closed and sterile environment of those single-use disposable cartridges. All the tubings and fluidics are inside those cartridges, so there's no carryover from sample to sample, no contamination due to the closed system, and also no aerosols are produced during that process, which is important for operator safety as well. So that being said, I also would like to mention that all our consumables are, in addition to the research use only versions, available in MAX GMP format for GMP-compliant cell sorting. And that is true for a sorting cartridge, for the running buffer, which is applied, as well as for a quite big portfolio on fluorescent antibodies for cellular labeling. And in the end, that means that all that touches the cells during the sorting process is provided in a GMP-compliant format, and therefore also comes with a very extensive documentation, such as a batch-specific certificate of analysis, a certificate of origin or TSE statement, including animal component-free statements, as well as a quite detailed product information file, which discusses different points such as a description of products, regulatory information on raw materials, but also specifications, for example, QC release testings or in-process controls, as well as safety information, for example, endotoxin testings, biocompatibility, sterility testings, as well as particle analysis. And with that whole package, this, of course, makes it much easier to translate into the clinical environment when dealing with regulatory authorities and saves a lot of work in terms of risk assessment. And with that, I would like to come to the second part of my presentation, and I'd like to give you a short application example from the field. And in the beginning, I just would like to remind you, why is it so important, or what is the benefit from the MACSQuant Tyto Cell Sorter in terms of clinical cell sorting? And that is that the sorting happens exclusively in the fully closed and sterile cartridge. There are actually no fluidics inside the instrument, and all consumables are also available in a MAX GMP grade. Currently, the Tyto is already used in 9 different running clinical trials, either in phase 1 or phase 2. Over 200 patient samples have already been sorted within those trials up to date, and over 50 patient infusions have been provided. And we are also expecting to expand into at least 3 additional trials this year. Now, what are typical applications we see customers in the clinical segment are currently using the Tyto for? Now, here's a list of the top 5 applications we see. Of course, it's not only limited to that, but those are ranging from antigen-specific T cells over Tregs, CAR T cells, as well as MSCs, as well as iPSC or iPSC-derived cells. And as we don't have time to go into too much detail for all of those applications, I would just like to give you one example, and I would like to focus on the iPSC or iPSC-derived cell types. In this case, I would like to mention a clinical trial currently being conducted at the Center of iPSC Cell Research and Application, the CIRA in Kyoto, Japan. Here, a first in-human clinical trial, so real pioneer work, is being conducted by using iPSC-derived dopaminergic neurons in order to treat patients suffering from Parkinson's disease. And this clinical trial is led by Jun Takahashi and his group. And once being asked what actually was the biggest challenge in translating this therapy into a clinical trial, he answered that it was the sorting of their target cells, in this case, corin-positive cells. And back then, they were still working with a FACS droplet sorter. When they learned about the MACSQuant Tyto technology, they were very interested and performed a head-to-head comparison using their protocol, and in the end compared the sorted cells in terms of sorting performance, which was actually very good for both of the systems and comparable, but also in terms of like how cellular functionality and viability was after the sorting process. And I think it's important to shortly show you the protocol they're actually applying in that clinical study. I don't want to go into detail here, but just like to mention that from the first part, so the establishment of the iPSC cell line, to the last part, the transplantation of the dopaminergic neurons, which have been differentiated back then, into the patients. They're actually injected into the brains of patients suffering from Parkinson's disease. There are 30 days in between. And on day 12, 13, the cell sorting step actually takes place. And as you can see on day 26, so 13 days after the sorting process, the QC is being conducted, and it is going to be decided whether the cells can be transplanted or not. And a first result I would like to show you is exactly from that QC date, so 13 days post-sorting. And what I would like to mention is that those dopaminergic neurons, they're actually forming spheres in the process of their differentiation. And the bigger the spheres, the better for the cells. So the higher the viability and functionality, and also the division rate, of course. And what you can see already by eye is that the sample sorted with the MACSQuant Tyto shows bigger spheres. This is significantly different compared to the FACS droplet sorter, and ends up in a cell yield of 160%, and is probably due to the very gentle sorting on the MACSQuant Tyto, leading to highly viable and functional cells. Now, once being transplanted, the cells, of course, have also a job to do. They have to grow, and they have to survive. And there's also a way to measure that in vitro, and that is actually by checking the area of neurite extensions of those cells. This is an indicative of graft survival capacity. And what you can again see by eye is that in the Tyto sorted sample, this area is bigger compared to the FACS droplet sorter. This is significantly different, and therefore we expect a higher graft survival capacity of those cells after they are transplanted. Another important part for clinical trials and clinical work is, of course, the time you need for your protocols. You can see here, using the FACS droplet sorter, the group of Jun Takahashi needed 18.5 hours to sort cells needed to treat one patient. We were able to cut that by 1/3 down to 12 hours using the MACSQuant Tyto, which is due to the fact that we had a higher final cell number in those samples. And of course, the group was very happy with cutting down this time. And in the end, they actually decided on bringing the MACSQuant Tyto Cell Sorter into that running clinical trial on treating Parkinson's disease using iPSC-derived dopaminergic neurons. In 2019, several patient samples already have been sorted successfully within this trial. And if you're interested in more detail, the data I presented and more were also shown in a poster presentation at ISSCR 2019, and you can get this information if you're interested in. And I think with that, it's time to end my presentation. I would like to thank you very much for your attention, and I'm really looking forward on the perspective of all the people here in the roundtable discussion on using multiparametric cell sorting in the clinical environment and on their specific applications. And I hope you enjoy the rest of this roundtable discussion. Thank you very much.
Thank you very much indeed, Felix, for a really interesting presentation. Very cool data there. So I would now like to invite our panelists to join today. So first is Dr. Stephen Ellerman, founding scientist and chief scientific officer at Upstream Therapeutics. In 2012, he conceived the EU Red Star program, which led to the first rounds of private investment for Upstream. And Upstream has since grown from 1 employee to a team of 18, and Steve has led the discovery, development, and translation of the proprietary CD360 MSC technology. He is joined today by Ivy Lay from MD Anderson Cancer Center, who has over 10 years' experience in adoptive T-cell therapy, working with Dr. Cassian Z. She specializes in and is responsible for training the next generation of scientists in clinical manufacturing, clinical-grade cell sorting, and T-cell generation for multiple clinical trials targeting various tumor sites. And our final panelist today is Dr. Jack Dunne, Senior Consultant at Miltenyi Biotec. Jack is a scientific and business executive at Miltenyi, formerly OWL Biomedical, and has acquired 30 years' experience in applications and instrument development in flow cytometry and imaging cell analysis. Most recently, his responsibilities focused on enabling novel cellular therapies by specific development and application of cell purification technologies, including the MACSQuant Tyto Cell Sorter. Well, thank you very much indeed, everyone, for joining me today. I'm very much looking forward to our discussion. Could I firstly ask our panelists, Stephen and Ivy, to each introduce us to your current cell therapy projects, and perhaps in particular to share with us what you've seen as the biggest challenges in the process of getting regulatory approval and bringing these into clinical trials. Ivy, we'll come to you first.
Hi, yes. Our lab specializes in generating antigen-specific T cells targeting multiple cancer sites. We start with the patient's T cells, which are very low frequency in the antigen of interest, so we have to sort these cells out from the patient's leukapheresis product after several rounds of stimulations before we can expand these cells to dose for patient infusion. Some of the challenges that I would say for getting regulatory approvals for getting these sorted cells into the clinic is the antibodies that are available that need to be GMP-grade, but because we're a phase 1/2 trial, it's very difficult to get reagents such as dextramers or tetramers that are GMP-grade for personalized therapy, especially for targets that are new to the industry or that we're researching new antigens that target multiple cancer sites. It's very difficult to get GMP-grade reagents for such personalized use.
Excellent, thank you. And Stephen, how about Upstream?
Hi, can everyone hear me okay?
Yes, absolutely, loud and clear.
Excellent. Hi, I'm Steve Ellerman. We start with two cell sources. We start with human bone marrow to isolate stromal cells and also umbilical cord tissue. Right now we have 4 phase 1/2 clinical trials running to develop the marrow-based drug for diabetic kidney disease, and we have the other 2 trials from the cord-based stromal cell, and that's running in autoimmune liver diseases, autoimmune hepatitis, and primary sclerosing cholangitis. And we also have a trial running now in moderate to severe ARDS with the cord-based drug. In terms of challenges, we have 2 major focuses. In addition to the GMP reagents that Ivy just highlighted. The first challenge we've really focused on was, if you like, sort of upstream preparation of the tissue, specifically the umbilical cord tissue. For those of you who don't know umbilical cord tissue, it's one of the toughest tissues known to humankind. And I imagine what we'll dig into during this call is the critical nature of upstream tissue processing for cell therapy. And we've been really focused as we've evolved our cell therapy isolation on improving that upstream processing. In the first instance, we started out with a MACSQuant-based process, and our upstream tissue processing was somewhat crude. And as we've enhanced the selection process with better antibodies and moving through FACS technologies and onto the Tyto, we've really had to hone that tissue processing. So one of the takeaways really is the challenges of upstream solid tissue processing, whether you're working with tumor samples or umbilical cord tissue. Secondly, one of the great challenges is open aseptic processing, which is fine for phase 1-2 trials, but as you move to phase 2B and phase 3 commercialization trials, open aseptic processing becomes, something that's cost inhibitory, and there is really a strong desire to move out of grade A, B type backgrounds into grade C backgrounds, and that would obviously lower the overall manufacturing costs because your clean room space is cheaper. So really being able to close out that front end of the process, the selection process, you really want to use a closed process, and the Tyto really has enabled us to start, thinking about doing sorting in a more grade C type background. So the big challenges for us have been closing the front end of that process, which is something we've been working with the Tyto to achieve. And then obviously upstream processing of that tissue, in addition to the antibody and reagent concerns that Ivy mentioned.
That's great. Thank you very much indeed. And Jack, how do these challenges resonate with you in the broader context of trends in cellular immunotherapy?
Thanks, Eliza. I think we have the luxury of enjoying comments from two very early pioneers in this kind of strategy of using cell sorters in a clinical lab. Of course, they're in a GMP environment, and there are others who've struggled with droplet sorting for years, and I applaud their effort. The interesting proposal I'd have is, of course, I think we all understand that the regulatory environment is evolving. Our regulators I think, have done a great job at trying to enable very important clinical studies for, discovery of efficacy, and I think it's gone well. I wonder how things will change as alternatives become available. So when there are no GMP reagents or closed instrumentation, I think there's been a lot of flexibility or hope or forbearance anyway. And I wonder how that's going to change now that Tyto is available and now that many GMP reagents are available. And so when you introduce new technology, I think that is likely to be reflected in changes in regulatory strategies that are going to be, forgiven or allowed. And that's, of course, that's a global thing. And I wonder how that variety is going to percolate around the world.
That's a very interesting point. Yeah, absolutely. So then looking at our next question, it'd be great to just do a little bit more of a deep dive, Stephen and Ivy, regarding how you previously approached cell isolation of target cells in the past and sort of what your learning curve has been. So Stephen, you touched upon this already, but it'd be great to hear a bit more from you here.
Yeah, so I mean, obviously when we started this, we really realized we had an antigen that was good for isolating stromal cells from multiple tissues. So we started where everybody starts, with the Miltenyi catalog. We started working with MACS-based reagents to purify, in the original setting, our antibody is CD362, Syndicam-2. So we developed an antibody to that, and we started working with MACS, and that was great. We got a 10-fold, 20-fold enrichment in both marrow tissues and umbilical cord tissues, and it was great. But what we were finding was, very much that we were, because of the way we process umbilical cord tissue, we were bringing quite a lot of, dead cells with us, and that was somewhat limiting our, if you like, sort of our growth potential in that early phase. So because the Tyto wasn't available, we moved to droplet type sorters, and we added a second antibody, and that really helped. Our purity went up, 30,000 fold. That's when we realized we were really onto something. And we started getting almost, every cell that we were sorting was a stromal cell, and we actually started to fractionate into 362 positive and 362 negative populations. So we started to see a real, increase in purity, but the yield still wasn't great. So being able to look at, the Tyto instrument, we realized that we were going to go clinical with this, and the desire was always going to be to maximize yield as well as purity. And one of the things we noticed early on with the Tyto, one of our key criteria, much like you heard with the iPS-derived dopaminergic neurons, what we found was one of our key criteria is colony-forming units of fibroblasts, CFUF. And what we saw was increased CFUF with the Tyto-based sorts. It was about almost twice as many cells were actually plating and forming colonies. And that was key for us, because it kind of suggested that the process was somewhat gentler, and we can expand on that later. But really, we did RNA-seq between droplet-sorted stromal cells and Tyto sorted, and the Tyto sorted cells were very much akin to unsorted cells. They didn't exhibit the same levels of, if you like, sort of HSPs and stress genes. So it's really been an evolutionary journey. Really, from the Benchtop open max process, all the way through to, moving through droplet sorting, getting the bones of a process in place, and then realizing that, that you really want to get a closed process, and moving to the Tyto gave us that, double benefit of being able to close out the front end of that process. And, as it stands today, we're getting more stromal cells, coming out of the cord because of the more gentle nature, the lower pressure of the Tyto sort.
Fantastic. And Ivy, do you sort of see a similar evolution?
Yes, I have. I agree with Stefan and Jack in that there's an evolution of the criteria that are constantly evolving because of the technology that is presented and how it evolves over time. Like Stefan, we also started our trials with a droplet-based sorter. We actually had to put a BD influx into the GMP facility when we were back at the Fred Hutchinson. We implemented one of the first studies that integrated clinical grade cell sorting in the GMP facility. We actually had to have a specialized flow specialist inside the GMP facility with us that is specialized in running the instrument, because a lot of these technicians, we can train them, but the instrument is so advanced that it's hard to manipulate and it's not very user-friendly. We had to actually change the tubing sets in between patients, and the setup of the instrument, especially the droplet sorter, was very difficult to maintain sterility and avoid cross-contamination between products. Because we had to change the tubing sets between patients, there was a long setup period in the beginning of each patient sort where we had to make sure the droplets were positioned correctly so that the droplets actually end up in the tube that we're collecting the cells from. The tubes were obviously open to the air, and the droplets drop into it, and we fill the tubes with media, hoping that the cells stay sterile, and we actually don't know on the back end until the cells are expanded. The sorts took up to 8 to 10 hours a day because of the long, extensive process of setting up the instrument, making sure the droplets are aligned correctly, and then actually performing the actual sort per patient. It makes a very long day. Once we received the Tyto, it shortened those days by a lot because we can now stain the cells in the biosafety cabinet, plug the closed system cartridge into the sorter, open up our sort profile, basically just press play, align our sort gates where our cells are, and then walk away from the sorter, which means we have time to set up the back end of the cell expansion where the cells will eventually go into after the sort is completed. So from 8 to 10 hours, we can now perform each patient sort in about 2.5 hours, which means we can sort multiple patients in any given day, whereas before we were restricted to maybe one patient a day.
Wow, that's a huge difference. Great, thank you. And so I guess, we've touched upon a little bit here, but I suppose what for you are the most important points regarding cell sorting for therapies intended for human use? Is it about these concerns of sterility and cross-contamination? Stephen, perhaps you could share with us your thoughts there first.
Yeah, I think it's multiple concerns as you move, from phase 1 to phase 2 and towards commercialization. Really, the driver's got to be on product quality. And obviously associated with that comes sterility, endotoxin, mycoplasma. But it's really about, as you evolve your process development, it's about, improving the critical quality attributes. You want to get better at making your product as you evolve from phase 1 with non-GMP reagents and max type reagents to a better, more refined process. So, and also, that's driven by your engagements, your regular engagements with your own government's regulators. So for us, the focus point has really been zooming in on, the purity of our product, the 362 selection, and maximizing yield. So my own primary view would be around, the primary quality attributes of the therapy itself and what allows you to maintain that. And then secondly, around, cost per manufacture. The goal really is to, maximize the value there. So again, coming back to my earlier points, really around closing and automating anything that allows you to close and automate your manufacturing process, anything that allows you to get out of that grade A/B environment and into a more, automated grade C type environment, that's going to reduce your manufacturing costs and bring down your COGS. So, those would be two of the key drivers. Obviously, Ivy's really also highlighted some key attributes of the Tyto that are helpful. The lack of cross-contamination, the ability to save your negative fraction, to save that fraction in case something goes wrong so that you can rerun it. Those are all admirable add-on attributes of the Tyto as well.
Fantastic. And Ivy, would you sort of agree with those priorities?
Yes, definitely. I think the sterility of the sample, being able to sort in a closed system is very important. Now that the FDA knows that other systems might be available, they're really questioning those things. We have to make sure that we can show that the system is sorting and that the cells are sterile. There's no cross-contamination between patients, and the Tyto really allowed that ease of use because of the single plastic cartridge where we can use it single use and then dispose of it between patients. There's also no cleaning of the instrument between patients, which allows us to sort multiple patients a day because it's in a closed system, which means there's no patient samples dropping out of the cartridge into the instrument, which means there's ease of use of time for setup, and the ability to plug and play and walk away from the system is very critical, as well as cell functionality and viability. Post-sort, we have to still have viable cells so that we can expand the cells down the line so that we can reach dose basically to infuse into patients. Cell functionality, we have to get a pure population of cells. The FDA has strict criteria now for us on how pure the sample is that we can infuse into the patient. They want to avoid any adverse events, obviously, so that the Tyto implementation really help us fight these challenges and implement that into clinical trials.
Fantastic, thank you. And obviously you mentioned there, the goal of sort of getting regulatory approval and working with the regulators. Could you share a little bit more details around your personal experiences in working with the various regulatory bodies, and has it been quite challenging in any particular areas in sort of navigating what's required from them? And perhaps, Ivy, we could sort of continue with you there.
Yeah, we definitely had to get regulatory approval, and initially we had to show that the cell sorter actually works. So to address the issues of sterility and not having any contamination of the samples, we placed the cells into a 2-week rapid expansion, and post rapid expansion, we had to make sure that we send for sterility, mycoplasma, endotoxin to prove that the product is actually sterile and safe to infuse into the patient. We also had to analyze the sorted samples, pre-sort, post-sort, and the waste analysis to analyze purity and yield of the product to make sure that we're infusing the purest samples that we can into a patient. There are a lot of testing that we needed to perform to ensure that the product is running consistently and smoothly as we implement this into our trials.
Great, thank you. And Stephen?
Yeah, I'd probably echo a lot of what Ivy has commented on there. Really, the regulators at these stages of the trials process, phase 1 and 2, they've been really, focused on, the sterility of the process in all aspects, mycoplasma, endotox, the reproducibility of the process, certainly, around, if you're going to do, 12 isolations, 15 isolations, in a month, how reproducible are they? So I would say, arguably the regulatory response has been overtly favorable to improvements in cell selection processes. The onus is really on developers to, bring forth the data that shows that, if they do make changes to their processes, as we've done, as we've gone from a max-based process to a Tyto, it's really about highlighting to the regulator the equivalence of the drug product that arises from the differences in the selection procedures that you actually use, and obviously again the Tyto has been favorably reviewed in the US and in the EU because of that ability to record that data as you go along, and the fact that it is closed and automated. I think as the Tyto moves through later stages, clinical trials, you'll see more robust regulatory examinations of the instrument. But so far everything's been very favorable.
Fantastic, thank you. And so then it would be great to chat to the panel regarding your perspective on future enabling technologies. Obviously, we can focus around cell sorting, but also maybe perhaps broader in the workflow. What needs to happen technology-wise to enable future therapies? Jack, we'll come to you for this first.
Thanks. Sure. Actually, a quick prior comment from a technology developer's point of view, as to drivers. I first, I've spent my career applauding and being amazed by droplet sorters. I kind of hate the idea of bashing such beautiful technology that's been such a great tool for all of us for over many years now. In fact, I'll comment that the droplet sorter patent goes back 50 years now. We've been riding that technology for a long time, and I think very successfully. Still, the drivers for a technology developer, a tool maker, is really to enable folks like Steve and Ivy and others who I think have done heroic work to try to validate the importance of flow cytometric purification of cells for therapeutic use. And it's been hard, really hard. A lot of folks have really struggled with this problem heroically, I think, and I applaud them. As a technology developer, it became clear to me that it was time to think of a new paradigm for sorting. And so when the microvalve technology became mature, I would say now 10 years ago now at Owl Biomedical, it became time to rethink the whole problem. And so all of these features and attributes that we've been discussing are all sort of sequelae from that natural transition from one beautiful technology to another. You couldn't have made this valve 50 years ago. We didn't have microfab technologies like we have now. So, as the world evolves, you need to periodically rethink your strategies. Now, so in terms of where we're headed, for me, the microvalve is still a new technology platform. The Tyto is the current implementation of the commercially available implementation of that platform. And I think there are lots of things that will be following surrounding this microvalve technology. It's all about keeping things closed. keeping things easy to use, all of these kind of regulatory benefits really come down to the notion of robust and reproducible performance. There's so much variability in our field with regard to, regulatory strategies, but more biologically relevant, variability with regard to patients, how they're going to respond to these various therapies, with regard to the cell preparations themselves. These are living medicine. They certainly are going to have their, complexities. And so it's key to control the manufacturing purification process, all aspects of it, whether it's genetic engineering or purification or any of the handling steps, even so far as the therapeutic delivery, whether it's allogeneic or autologous, all of this chain of workflow from initial all the way to infusion, from isolation to infusion, all of those things need to be controlled. And I think the microvalve is a key component in that whole chain of sample of cellular therapeutic development and delivery. So I guess that's where I would head, and maybe that's an interesting discussion, or I would encourage questions from the crowd about other steps in the process that, of course, Miltenyi is clearly focused on enabling cell therapy broadly. And so I'm anxious to hear more as a technologist about other steps. I kind of think we solved this one. Maybe that's a little grand and maybe premature. That's okay. I hope you'll forgive me. But there are other pieces that really need our focus, and I hope we'll get to them soon.
Fantastic. Thank you. Stephen, I wonder if you could sort of share your perspective there on what you think is where the greatest need might be.
How do I follow Jack?
It's tough.
Yeah, I mean, I'm like sort of my own view is probably very very similar to Jack's, and it's kind of driven by what Ivy said earlier. I mean, to take this field forward, we need walk away technologies. Right now, when my guys, my team, and they're the geniuses here, they go into a 362 selection. The upfront processing takes time, the sort takes time. And yes, we're reducing that time with technologies like Tyto, but developing, automated, self-aware technologies that allow you to, process tissues automatically, get a uniform blend, load that onto a Tyto, have that run and store the cell. Almost push button, as Ivy was talking to, being able to automate those processes and have it record as it goes, making it autonomous. I think that's where certainly cell therapies, because of the potential benefits and because of the bespoke nature of a lot of the cell therapies. I think being able to you'll hear it a lot in the field, but industrialize it, and the only way you can do that is by automating. So I think we're getting there with cell expansion technologies, but I think that's where that's really where the upstream processing needs to go, to allow us to have that automated, so that you are selecting and enriching and doing this upfront work less manually, which is where the science and the expertise is. As Ivy spoke to, when we started out with droplet sorters, it really was you needed a bona fide flow cytometrist to help you set up, the technology, and taking that into a clean room was a big challenge. Tyto is simplifying it, and hopefully Tyto and associated technologies will continue to simplify it so it can become more prevalent. So that would be my take. If there is going to be a push, we're seeing it in downstream expansion technologies, but in the upstream, really industrializing it and automating it so that we can run, a dozen, 2 dozen, 3 dozen selections all at the same time.
Yeah, absolutely. And Ivy, do you sort of see any additional gaps there in the current manufacturing workflow where you think there's a technology need?
I do. I mean, I think that right now when we're sorting cells, we actually have to QC the cells post-sort just to check for purity, because the instrument doesn't tell us what the purity is post-sort. I mean, we can guess that we're gating on the correct cells, but the instrument is not, visually telling us how pure the sample is post-sort. So we have to take a sample out and run it on another cytometer just to check before expansion, so that we're sure after the 2-week expansion that the patient will be ready for treatment and we can proceed with treatment as planned. So I know that Miltenyi is working on a self-aware Tyto that maybe does this in the instrument while the cells are sorting, which would be very helpful. I mean, I know that the sorters are being upgraded constantly. Another thing is maybe like what Stephen had said, doing multi-parameter sorts in one cartridge at the same time, so that we're collecting different cell phenotypes in different chambers, or starting out with the ability to start out with a larger volume of starting material. So maybe we can eliminate one of the expansion phases where we get the cell numbers that we need to infuse directly into the patient, to eliminate the time that it takes for manipulation and expansion, so that we can eliminate a long, 8-week process to 4 weeks or whatnot. The ability to maybe deplete and enrich a population before you sort the cells, so that your starting material is enriched so that sorting would be easier, or maybe coming up with commercially available GMP-grade reagents that will pass FDA approvals for cell sorting for personalized therapy.
Yeah, very interesting point there. Thank you. So I'm conscious of the time, but we have lots of questions that have come in from the audience, so I will just crack on with those now. First of all, Jack, one audience member would like to know if you have any experience in sorting human neutrophils or monocytes with the Tyto technology, as these cells are usually very sensitive to manipulation and droplet sorting.
So we certainly do, and in fact, I'd encourage you to go to the Miltenyi website, and you can see some app notes with regard to this. But I would also caution that what we found over the space of some years now is that the differences with regard to the gentle aspect and the kinds of metrics for, are the cells happy, if you can use the archaic cell culture term, these are subtle effects, and they can be critical, as in the IPS story just described, or Steve's story, or Ivy's. But I would encourage you to look at your own requirements in a very particular way. So it's easy for us to say it seems gentle, but I think the key is going to be to look at your, Steve would say the quality metrics. What do you care most about as with regard to how the Tyto would give you cells that are different from a droplet sorter or a magnetic sorter or a elutriator, or there's lots of purification platforms. So I'd say sure, we've done it and it works great, and you can look at the data, and I would say you need to try it and see if it matters for you. Yeah, great advice, definitely.
And we've had quite a few questions coming in from the audience that sort of pertain to using this sorter instead of the CliniMax, or using this in combination with other platforms. I was wondering, perhaps Ivy, if you had any sort of experience to share there in terms of the technology that you've utilized.
Yeah, we actually did use the CliniMax and did like a CD25 depletion or a CD8 selection prior to sorting out our antigen-specific T cells. I don't think I would replace the CliniMax Plus or replace the CliniMax with the cell sorter, but it really depends on the end result and what kind of cells you're trying to sort out. I mean, it also depends on the number of cells that you require post-sort. Obviously, you can sort and select for cells with the Tyto, but I think the CliniMax might be for more of a large-scale enrichment, where you want to do it in like bags of blood, or you want to enrich bags of leukapheresis product for a certain specific type of cell and then sort post-stream. So it depends on the purity of the population you want afterwards.
Yeah, that makes sense.
Could I add a comment? For years, flow cytometrists have known that magnetic pre-enrichment and cytometric sorting just fit together beautifully. There's a lot of advantages that are complementary to the two strategies, and I really encourage you to think not either or, but and of course, as Ivy mentions, it's all about the numbers, and of course, there's detail about workflow and upsides and downsides for any step in a process. But I really think they fit together, and that's the beauty of being at Miltenyi, is that we're happy however our platforms enable. That's our goal.
Yeah, absolutely. Great. Another question from the audience. From a practical standpoint, does the instrument clog while sorting, and how do you recover from a clog event? Do you need to transfer the cells to a new sorting cartridge and continue? I wasn't sure if either, Jack, if you'd like to take this one for us.
Well, I'll start, but I know I want to hear Steve's response to this too, because I know it's been an important property for him. So the first thing I'd say, sure, yeah, you can clog a cartridge for sure. What we didn't describe is that, in fact, at the bottom of the input bowl, there's a 3D filter which catches big chunks of stuff or aggregates of cells. So we protect our valve from really substantial debris using this 3D filter that's at the bottom of the input bowl. The good news is that when you have a clog, it's not a catastrophe. You don't lose your sample. The instrument says, "Hey, I'm not working. Come help me." And we're working on much better mitigation strategies than that now. But your sample is all still there. You can harvest whatever sorts that you've done before the clog, and you can leave them in you can move them to another cartridge and pool. You can harvest the input that hasn't gone through the sorter yet and put it in a new cartridge. Yeah. So the mitigation, yes, is just use a new cartridge. And then of course, you start doing that often enough, and per Steve's comment, you start thinking more about upstream sample processing to pre-filter, or and this is another case where a magnetic column does a great job at getting rid of all this kind of stuff. These bead columns are great at trapping stuff, including cell debris. Non-specific binding to columns is a great way to clean up a prep.
Yeah. Great. And just quickly, Stephen, did you have any clogging stories you can share?
Do we have another hour? I'd like to think that all of the anti-clogging technologies that Miltenyi has developed on the Tyto are in some no small part to the collaboration with Upstream. The first things first. Yes, any sorter will clog, and I would say we have probably the toughest tissue to sort from in umbilical cord tissue. It's concrete covered in jelly, covered in glue. And it kind of has all sorts of problems. So I'd say we've clogged more cartridges than everyone else on the planet combined. The focus, really, all joking aside, is you're focusing on your upstream process development, so you understand that you have a sortable processed material is key. Obviously, I don't want to go too much into the specifics of our own. But we had one, umbilical cord processing protocol that worked for MACS. You can imagine that's quite straightforward. It's just got to use gravity to go across a column. And the desire to have a closed automated process and a more purified process on the Tyto, it has allowed us to, evolve that process and improve the, if you like, the enzyme complexes that we use to digest the cord. So the key attribute there is, as you're doing that process development, you lose nothing. As Jack, highlighted, if you're doing this on a droplet sorter and you're evolving your process dev, and the sample that you want goes into the wrong tube, then it's gone. It's gone into the negative fraction. It's gone and lost forever. Here you lose nothing. Everything goes into either the positive or the negative pool. So you can always just take the cartridge out, take your sample out, and resort. And we have you've done that for quite some time now with the optimized upstream process. And obviously there's newer cartridge technologies that the guys are developing that I may or may not speak to, but that's really improved things as well. So right now we've gone from sorting possibly one of the most complex tissues known to humankind to a 2-hour sort window where we're sorting 20-30 million cord tissue mononuclear cells down to 3,000 or 4,000 pure cells in 2 hours with no clog. So yes, sorts clog, but you take responsibility for that and improve your process dev.
Absolutely, great advice. And just a quick comment following on from Jack's response from Sandy Pelletier, our sadly absent final panelist, who unfortunately was taken a little ill. But thank you, Sandy, for watching today. She said that she found that cycle is necessary to sort tumour cell suspensions, especially the tumours that are necrotic. Wishing you a very speedy recovery, Sandy. So I'm afraid we have run out of time today, but thank you so much indeed to our panel, Stephen, Ivy, Jack and Felix,